All right, welcome to our lecture on the cell cycle. This is our last notes packet for Unit 4. So you should have the new notes packet, and here we go. You see this large diagram right here in your notes packet, and we are essentially going to label this diagram and kind of write our notes almost like we're annotating the diagram as we talk about the cell cycle. So first, sort of next to kind of at the top of your notes, next to the word cell cycle, let's make sure that we understand this is the life cycle of a cell. So we'll see where cells are born, how they grow and mature, even how they divide, and then how they can be triggered to die as well. The big cell cycle is broken down into two parts, and those parts are number one, which is the majority of this diagram, this outer kind of bluish green loop. And then here is the second, which has been labeled as 7. So I would like for you to label, we're going to talk about the majority thing right here. What is number 1? Next to the word number 1, I want you to write interphase. That is the majority of a cell's life. An interphase itself is broken down. You can see what this space right here holds are sections 2, 3, and 4. So it's broken down into 3 regions or 3 phases itself. But interphase is the first primary phase of the cell cycle. Now, this is going to account for about 90% of the life of a cell. And so the majority of the life of a cell, as we'll find here in interphase, is going to be about growth and getting itself ready for cell division. So we're going to find both growth of the cell and DNA duplication here in interphase. So at this point in your notes, you should have labeled number 1. So come to your diagram where there's a number 1, write the word interphase up here so you know that number 1 is interphase, and then you've written a few little notes about interphase below the word itself. Now let's talk about the 3 phases of interphase. So the first part of interphase, which is number 2 on this diagram, and it's fine to write in this space right here where number 2 is located, this is called G1. So label on your diagram where you see number 2, label this as G1. And then write a couple of notes about G1. G1 is the first growth phase of interphase. And really what we're focusing on here is growing the cell in size and in organelles. Because remember, the cell is going to eventually become 2 cells, split into 2 cells. So we need more stuff, right? So G1, or growth phase 1, is going to work to increase cytoplasm and also to duplicate organelles. Then we see what is number 3 on the diagram. This is the next part of interphase. It's called synthesis, or the S phase. The big thing that's happening here in the S phase, if you synthesize something, you are making something. And in this case, what we are making is more DNA. So we are duplicating our DNA in the S part of interphase. And then the last part of interphase right here is labeled number 4 on your diagram. This is a second growth phase. It's called G2. So in this growth phase, we're going to finish any sort of internal growth and development. And specifically, we're going to create something called the spindle. And the spindle is something that we'll address in our notes in just a couple of minutes. And so all of this on your diagram should be labeled interphase, and then the 3 parts of interphase, G1, S, and G2. If you'd like to see what that looks like, here is that diagram labeled interphase, G1, S, and G2. So the majority of the life of a cell is interphase. The second part of the life of a cell is short, and here it is seen. It's called the M phase. So let's talk about the M phase, the second part of the cell cycle. So in number 7, you should label that as the M phase. This is a very short portion of the cell's life, but the cell's very active here. It's going to actually divide. And so M phase itself is broken down into 2 steps, and you can see those labeled as 5 and 6. And they're really skinny pieces of the pie, so you might want to label them and then drop below the diagram and write about them. So the 2 parts that we see as 5 and 6, number 5 is called nuclear division. And this is either going to be the process of mitosis, which we're getting ready to talk about, or it's going to be meiosis, which we'll see in the next unit. But number 5 is nuclear division. We are literally dividing the cell. Number 6 is called cytoplasmic division or cytokinesis, and we are going to split the cell into 2 separate cells. So going back to that diagram, what they have labeled is mitosis. We know that that is nuclear division, and what they have labeled as cytokinesis is cytoplasmic division. Now, there's something that is a part of the cell cycle that's not actually on your diagram. So we are going to add a phase called G0. So I know this diagram doesn't exactly look like yours, but find in your diagram the section that is labeled G1, and you're going to kind of create a little arrow, an offshoot off of the main diagram, and label it as G0. So let's talk about G0 as an offshoot from G1. This is a quiet or resting phase that some, not all, but some cells exist. They can exist for their whole life in G0, just hanging out. They're metabolically active and alive, but they're not actively growing and working their way towards the M phase, division. And so you might see this word quiescence related to it. Think about like quiet. It's a very calm state where we don't see a ton of active growth. So remember, the cells are still alive. They're just not actively growing or duplicating anything here. And the last 2 bullets are a reminder that some of our cells live in G0 for their whole life. Examples, very common examples of cells that exist in G0, which is technically a non-dividing state. Muscle cells, skeletal muscle cells specifically, and then nerve cells, which you maybe know or have heard that once a nerve cell has been destroyed, you can't regenerate it. That's part of why it exists. It lives its life in G0. Some cells can enter and exit G0 as needed. So a great example of that would be our liver cells. They exist in G0 until maybe the liver cells are damaged or destroyed. And then they will hop out of G0, enter the cell cycle, grow and divide to replace those lost cells and re-enter G0 and sort of hang out there. It's a great energy efficient state. It is allowing our cells to continue to exist without putting a ton of effort and energy towards actively growing and dividing them. So G0 needs to be added to your diagram. Next section of our notes, let's talk about why cells even exist within a cycle. So keep in mind that the cell cycle, when we talk about that diagram that we just labeled, this is only occurring in eukaryotic cells. So I've given you a picture of a prokaryotic cell. So here's a bacterial cell. They don't do what we just talked about in that diagram. They do a different process called binary fission. It's very simple. We talked about it a little bit when we talked about the difference between prokaryotic cells and eukaryotic cells, but I wanted to remind you that everything that we're discussing is really exclusive to eukaryotic cells. That's plants, animals, protists, and fungal cells. So in eukaryotes, let's talk about the cell cycle in a single-celled organism versus a multicellular organism. Single-celled eukaryotes pretty much utilize the cell cycle, growth and cell division, to reproduce. They're going to make copies of themselves, and this is how they grow as a population. They're using the cell cycle for reproduction. But in more complex multicellular eukaryotes, we see that the cell cycle is used to repair. So anytime we have, like this image is showing, anytime we have injury to tissue, we will see mitosis kick in and repair and replace the damaged cells. We also use the cell cycle to reproduce. We can reproduce actual individual cells, and then we can reproduce as organisms, which is more about meiosis, and we'll get into that in our next unit. But as an organism, we grow. And so look at this last picture. As a baby, you are much larger today than you were when you were a baby. You have grown as an organism. You have developed as an organism. That is the product of your cells going through the cell cycle and dividing themselves. I mean, you are a larger organism today than when you were born because of cell division. So lots of different reasons in eukaryotic cells, especially multicellular eukaryotes like us, lots of reasons that we would utilize the cell cycle. Let's talk about some vocabulary that we're going to use a lot in talking about mitosis specifically, that is worth defining. Okay, so let's talk about a duplicated chromosome. So we know that DNA is duplicated in the cell cycle, specifically in the S phase of the cell cycle. That means that we end up with a whole lot of DNA, and it's kind of messy. There's a bunch of it, it's all tangled up. Think of a big giant mess of clothes in your room, just all piled all over the floor. The better and more efficient way to kind of deal with this stuff, this DNA, is to organize it. And so organizing DNA happens by kind of tidying it up, compacting it. And so if we take a look at this diagram right here, this is DNA. DNA will wind itself around these little tiny proteins called histones, and they're sticky proteins. And so DNA first will start to coil itself up around these histone proteins, and the proteins then start to stick together. And so we see the first visible structure of DNA under a microscope referred to as chromatin. And really at this state, it's already DNA that's been compacted a little bit, organized a little bit. Well, this chromatin continues to condense itself and stick together, and it eventually forms these structures that we recognize down here as chromosomes. And so when we look at particularly the M phase, mitosis and meiosis, we're going to see chromosomes. Chromosomes are really just a bunch of very neatly compacted DNA. And it's compacted for a reason. It helps, if we kind of organize it and compact it into these structures, we can make sure that when we actually divide the cell, that we get equal amounts and correct amounts of DNA in each cell after the division process. So if DNA were to condense prior to the if a chromosome were to condense prior to the S phase, it would look like just one little single stick, and we would call this a chromosome. If we look at what a chromosome looks like after S phase has occurred and the DNA has duplicated, the chromosome looks like it's duplicated, and it takes on this little X-shaped structure. So if DNA is duplicated after the S phase, it looks like an X. If the chromosome is created after the S phase, it looks like an X. If the chromosome is created before the S phase, which it rarely does, it would look like a little single stick. And so I want to continue in this section just to give you a last little bit of information. A duplicated chromosome, so something that looks like an X that's already been through the S phase of interphase. We still call it a chromosome, but it is now made of two parts. So here's one side and here's the other, and they are referred to as sister chromatids. So make sure that you label on the diagram in your notes, you label that each side is called a sister chromatid, and collectively we would refer to it as a chromosome. We still call it a chromosome. And then there's this little sticky thing in the middle holding the sisters together, and that is called a centromere. So that language is pretty important as we look at the steps and stages of mitosis. Oh, yep, sticky binding here holding the sisters together. The other thing that I want to talk about is something called the spindle. So you have this diagram in your notes, and the spindle is formed during G2 of interphase, and it's really important in pulling the sister chromatids apart once we actually start to talk about nuclear division in the M phase. And so within the spindle, there are a couple of things that I want you to label. The first thing that I want you to label is called the centrosome. And so over here on your diagram, you see this entire green area. It has a darkened center, and it's got these little green tubes radiating out. I want you to circle this whole thing that is green and label it as the centrosome. Within the centrosome, we see organelles. They are called centrioles. Those are the two little tubes that you see. So label these centrioles. As a reminder, these are only found in animal cells. That was something unique to animal cells, and we talked about it back in our organelles unit. So within the centrosome, there are centrioles. And then there are also these little fibers extending out. So some of the green fibers are called aster fibers. And then what we see here as green and purple are both going to be referred to as microtubules. So these are going to extend from the centrioles out to the chromosomes that have been aligned in the middle of the cell. And really, they are going to work in separating, pulling these chromosomes apart to opposite ends of the cell. So there's a centrosome on one side of the cell, there's another centrosome on the opposite side of the cell. They extend their microtubules to the middle and connect to chromosomes. And when the spindle starts to operate, it's going to pull those chromosomes apart. Now, the last thing to note here are what are called kinetochores. These are, and you can label them, these are the little purple dark circles on either side of the chromosome. These are going to act as connection points for the microtubules to hook in. And so it's almost like fishing, right? So back here, imagine if this is you holding your pole and you cast your fishing line out, you hook a chromosome, and then you reel it back into you. And so when you do that, it's really important that these sister chromatids separate so that you only pull half of each chromosome back to you instead of the whole thing. And we'll talk about issues, diseases, disorders that might occur if you accidentally pulled the whole big X chromosome structure instead of just half of it back to one side. So knowing the spindle now, I want us to watch an animation that is the process of mitosis. So check this out before we write down our steps. So this is entering the cell. Those are organelles. Now we're heading into the nucleus, and we're going to watch nuclear division. So we see some chromosomes form. We see the nucleus disappear, and here comes our spindle. It's going to attach out to our chromosomes, and then it's going to pull those chromosomes or those chromatids, the sister chromatids apart, reform the nucleus around each set of DNA, and pinch the cell into two separate cells. And we get cell division. Now let me check something right here. Yep, we're still recording. Okay, so let's go back into our notes and we're going to finish this up. You see this diagram right here at the top of your mitosis chart. I want for you to, above the diagram, I want for you to write a big picture. So as we look at this, the animation that we just saw is this picture flow. So we started with one cell that has a full set of chromosomes. Now that cell is considered to be a diploid cell, meaning that it has a full set of chromosomes, some from mom and some from dad, that was given to the organism during reproduction. We'll come back to 2N and diploid, but just so we know, we start with a full set of chromosomes. We're going to copy the DNA and then work to split the DNA and the cells and end up with two identical cells that are still full counts of DNA. So that's the big picture as we work through the chart. Keep in mind, this is an identical copying process. We're going to make a copy of a cell. Interphase. In your interphase chart, we know interphase is all about cell growth. We have to grow the cell in size and organelles, and we have to duplicate the DNA. This is going to set up nuclear division, an opportunity for nuclear division. Remember, most of the life of a cell is spent here in interphase. So here are some pictures of interphase. You can see we still have a nuclear membrane. We can't really see chromosome structures, but we're starting to see little like threads. That's our chromatin. And we see our duplicated organelles, specifically the centrioles, which are going to help in the spindle. This is a real life picture under a microscope of interphase in a plant cell. So here's the border of the plant cell. Here's the nucleus, and you can start to see some of that DNA beginning to condense. Next, we enter the M phase, active nuclear division. And so the first step in the M phase is prophase, followed by something called prometa. And so we're going to talk about both of those together, and you can write about them both in your chart. You can see here's prophase, and then here's prometa, which is just a little further developed in that process. Here it is in real life in fish blastula cells. Here's prophase, and then an extension into prometa. So what are we seeing here? We see that DNA begins to condense into structures that we can see under a microscope. And we see that our centrioles within the overall structure of the spindle are going to move to opposite sides. So we're getting our spindle fully prepared and formed. As we migrate into prometa, we can see that that spindle actually is formed now. And we can see as we migrate into prometa that we don't have a nuclear membrane. It breaks down so that the chromosomes can occupy the full middle of the cell for an organized division process. Next, we move into metaphase. And so here's a cartoony version of metaphase. Here's real life metaphase. You can see that the sister chromatids in their chromosome structure are all lined up in the middle of the cell. That's called the equator of the cell in a single file line. This is the longest phase of all of the M phase portion of the cell cycle. And we're going to make sure that the spindle, not only do we line our chromosomes up in the middle, we make sure that the spindle is fully attached to each chromosome. Once metaphase is done, anaphase is triggered. And so anaphase, cartoon version, real life version, anaphase we see is marked by the separation of the sister chromatids. They are pulled apart now, and they're being pulled back to their centrosomes. The chromatids take on this V shape as they're being pulled apart. So you can see the V shape really well in this diagram. You can see it a little bit here in this diagram. This is the shortest of all of the M phase stages. And then we wrap with telophase. So this ends nuclear division. Telophase is going to basically reverse all of prophase. So we're going to see that the spindle is going to dissolve, that the nucleus will reform around both sets of chromosomes now at either end, and the DNA is going to start to uncoil and sort of relax itself out of its chromosome shape. Now that wraps up nuclear division, but that's not the only part of M phase. Remember, we have nuclear division and then we have cytoplasmic division. So the last thing to talk about in your chart is cytokinesis, right? And so this is what's happening when the cell is actually separated into two cells. And the process looks a little bit different in animal cells compared to plant cells, mostly because plant cells have this rigid cell wall. And so it's hard to pinch them in half. So in an animal cell, we're going to see the formation of something called a cleavage furrow, which is this pinching here in the middle that's showing the beginning of the separation between the two cells. The cleavage furrow is created by a ring of actin proteins that act like a rubber band. And so it's called a contractile ring, but it's basically going to tighten until the two cells are separated. In a plant cell, that doesn't work. So instead of pinching the two in half, in a plant cell, you see the formation of what's called a cell plate, and that's this little dashed line right here. So cellulose is going to be deposited right here in the middle of the cell until it actually forms into a new cell wall, and that one cell will now be separated into two cells. I know that was a lot, but we needed to work ourselves through the process of mitosis so that we are ready for our next time in class together. So once you're done with this lecture, you can be done with this section of notes.